All right, so let's talk a little bit about Chapter 8. That's going to be microbial genetics. Hang on just a second. Let me adjust my camera just a little bit. Oh, righty then. Okay, so we're going to talk about microbial genetics. Genetics is the study of inheritance, or commonly referred to as heredity, of living things. It is a wide range of science, lots of different fields in genetics that you can study if you wanted to become a geneticist, for instance. Among some of the things are the transmission of biological traits from parent to offspring. You could possibly go into a field where you counsel parents who have some underlying genetic issues, like maybe they have a relative that has had a child with Down syndrome and they want to know what the possibility of them having a child with that condition might be, how these traits are expressed in an organism, what is it about my genetics that made me, back when I had hair, to have brown hair and green eyes, the structure and function of genetic material, how is this genetic material assembled, and what does it actually do, and then how this material changes over time. The genome is the sum of all the genetic material in an organ, or an organism, excuse me. You can look on the internet, you can see things about the Human Genome Project, where the scientists took the human genetic code and have identified the different genes that are work about or that are responsible for different jobs within the human organism. Most of the genome exists in the form of structures called chromosomes. When we talk about chromosomal material, organisms go all the way from having a single chromosome. Most bacteria, for instance, have a single chromosome, all the way up to human beings have 46 chromosomes, and there are other animals with even more, and plants with even more. Some genetic material appears as a structure called plastids, or in certain organelles of eukaryotes. Plastids, see if I can get the little pointer to work. Plastids appear in bacteria, for instance. We will talk a little bit more about plastids later on in the summer or in the semester. And then certain organelles in eukaryotes. You and I have a structure called a mitochondria that has its own DNA. Plants have mitochondria. Some plants also have an additional structure called a chloroplast. And we are discovering now that these chloroplasts also have a unique DNA. The genome of the cell is composed exclusively of DNA, where the genome of viruses will contain either DNA or RNA. We had a chapter on viruses earlier, and we talked about the difference between DNA and RNA viruses. A chromosome is a distinct cellular structure composed of neatly packaged DNA. For eukaryotic organisms, the DNA is wound around a protein called a histone. The DNA is located in the nucleus. They can appear either what is referred to as diploid, as in pairs, or haploids, single chromosomes. At some point, cells in the human organism are considered diploid cells. I said earlier, humans have 46 chromosomes. We actually have 23 pairs of chromosomes. We have a chromosome 1 we got from our dad, and a chromosome 1 we got from our mom. A chromosome 2 from dad, a chromosome 2 from mom. So we got 23 pairs. In bacterial organisms, the ones with a single chromosome, that is considered to be a haploid cell, and that one, the chromosome is duplicated and transferred from one generation to the next through a process called binary fission. In most eukaryotic chromosomes, they are more or less linear. Bacterial chromosomes, however, the DNA is condensed into a package by means of a histone-like protein. Bacteria can have 1, 2, and sometimes several chromosomes. Again, generally, bacteria are only going to have 1 chromosome. They will sometimes have a chromosome and a plasmid, but there are bacteria that have multiple chromosomes. The difference between this is, and this doesn't show it very well, it doesn't show it at all actually, but while eukaryotic chromosomes are generally linear in appearance, prokaryotic chromosomes are usually circular in appearance. There's 3 different categories of genes. There are structural genes. Those are the genes that code for proteins. These genes produce the RNA machinery used in protein production. So it's the gene that is responsible for making that messenger RNA that eventually becomes a protein. Regulatory genes that control gene expression. In regulatory genes, you have something called the genotype and the phenotype. Hopefully in your biology class, you were instructed the difference between a genotype and a phenotype. A genotype is the genetic makeup. That's the sum of all the gene types. It's the distinct genetic makeup of a gene, or excuse me, of an organism. The phenotype is the expression of those genotypes or the physical appearance of the genes. These are responsible for the structures or functions of those genes. So if we think about eye color and remembering eye color, if you learned this in your biology class, eye color is the result of several different genes working together. But just as a for instance, we will say you get 2 chromosomes or you get 2 alleles for eye color. You get one from your mom, and say you get a big B from mom and a big B from dad, and that means your phenotype then is going to be brown eyes. Your genotype would be big B, big B. Your phenotype would be brown eyes. So the phenotype, the physical expression of those genes. The base of the DNA structure is a structure called the nucleotide. The nucleotide is a molecule made up of 3 parts. There is a phosphate group. There is, in DNA, there is a sugar called deoxyribose sugar. And then there is a nitrogenous base. And there are 4 different nitrogenous bases associated with DNA. And we will talk about those in a few minutes. These nucleotides will covalently bond to each other in a sugar-phosphate bond. This is going to become the backbone of the DNA strand. Each of these sugars is going to attach in a repetitive pattern with 2 phosphates. One of the bonds is to the number 5 sugar, or 5 carbon within the sugar. On the deoxyribose, the other is to the 3 carbon. That's where, in a moment, we're going to talk about one runs from 5' to 3'. The other one runs from the opposite direction, from 3' to 5'. And we'll show a picture of that here in just a moment. Nitrogenous bases are going to attach along the strand by covalent bond at the 1' position of the sugar. 2 groups of bases. You have the purines and the pyrimidines. They join with the complementary bases using a weak hydrogen bond. Molecules can be easily unzipped to gain access to the information encoded. If not for these very weak bonds, the molecule would be much more difficult to open, would require more energy, and things would not be as efficient as they are. Thankfully, these hydrogen bonds allow for DNA to be very easily opened, and these hydrogen bonds also reform very easily. So it doesn't take a lot of energy to open the DNA up, to expose the genes so the messenger RNAs can be made, but it also doesn't take much energy then to close them and bring it all back together. The base pairs will dictate the formation of the hydrogen bond. Adenine will always form a bond with thymine. Guanine will always form a pair with cytosine. So A always forms with T, C always forms with G. They have what is called an antiparallel arrangement, where one side runs in one direction, the other side runs in the opposite direction. So again, one side runs from 5' to 3', the other side runs from 3' to 5'. This plays a significant role in the way DNA is produced. If they both ran the same direction, there would be a lot of congestion in the area where the DNA was trying to replicate. But since they run in opposite directions, that frees up movement and allows for a much more efficient way of doing things. Overall, DNA replication is what is referred to as a semi-conservative replication. It is the careful orchestration of the action of 30 different enzymes. All at the same time, these enzymes are separating the strands of the existing DNA, copies one strand, and produces two complete new daughter strands or two daughter molecules. Each daughter molecule is identical to the parent in composition. However, neither one is completely new. One strand or one side of the DNA serves as a template, which is the original pattern of the DNA. The other side is the new information. So, a quick rundown of some of these enzymes. And again, I will not ask you to remember the function of all of these enzymes. But the helicases unzip the DNA helix, so its name kind of tells you what it does. The gyrase helps to untangle the DNA supercoils. If you think of, have you ever seen a child's toy called a gyroscope? It's something that spins sort of within itself. It's a top within a top. So you get that untangling or that coiling. Gyrase. Primase synthesizes the RNA primer. DNA polymerase 3 adds the bases to the new DNA chain and also proofreads, looking for mistakes. DNA polymerase 1 removes the primers, closes gaps, and repairs mismatches. Ligase is the final binding of the DNA during synthesis and repair. As you will notice in a moment, I'm sure there's I'm sure there's going to be a picture somewhere that's going to show that as DNA is being made, one side is not made in one long continuous strip. It's made in these little short batches. These short batches are actually called Okazaki fragments, and it is the job of ligase to attach all of those Okazaki fragments together. And then topoisomerase 1 and 2, supercoiling and untangling, again, they help the gyrase do its job. DNA replication, we will let you read that on your own, but again, this is the story of DNA. Daughter molecules, elongation and termination. Some bacteria will add about 750 bases per second to each of the new nucleotides. DNA polymerase 1 is going to remove the RNA primer, replaces them with new DNA. The ligases move along those lagging strands, links the fragments together, and completes the synthesis and separation of the two new circular daughter molecules. Mistakes happen in DNA replication at a rate of approximately 10 to the 9th or 10 to the 10th times. That would be a billion times. So there's like one mistake every billion or so bases. Transcription and translation you should be familiar with. Transcription is the master code of DNA is first used to synthesize a messenger RNA molecule. During translation, that messenger RNA is used to make a protein. There are a few exceptions to the transcription, to the DNA makes RNA, RNA makes protein. There are some RNA viruses that can convert their RNA to another RNA. And there are some retroviruses that convert their RNA to DNA. And there is a wide variety of different RNAs used to regulate gene function. RNase is similar to DNA in terms of its general properties, but its structure is different in several ways. While DNA is double-stranded, RNA is single-stranded. It exists in a helical form. It is a single coil rather than being a double coil. RNA can form secondary and tertiary levels of complexity, leading to specialized forms of RNA. If you think back to protein formation, when we talked about that, there were secondary and tertiary protein structures. RNA contains a base called uracil, signified or initialed by the capital letter U. There is no thymine in RNA. However, this does not change the DNA code because uracil follows the same pairing rules as thymine does. It contains ribose sugar, therefore it is referred to as RNA, as opposed to DNA, which contains deoxyribose sugar. Some varieties of different RNAs. There are microRNAs. These are little small pieces of RNA found within certain organisms. There's antisense RNA. There's ribose switches, and there's small interfering RNAs. All of these play a role in protein production as well as protein and DNA regulation. Primer RNAs are operative in both bacterial and eukaryotic cells. We will later on in the semester, we'll talk about some regulating things, and we will talk about primers that start the process to happening. Those primers are short pieces of RNA. And ribozymes, which are enzymes made of RNA. They remove unneeded sequences from other RNAs. Sometimes when the RNA is made, there's more to it than actually needs to be there, and it's the job of ribozymes to remove that unneeded portion. So here's 3 RNAs involved in transcription. There's transfer RNA. This is the one that's actually going to bring the amino acid to the growing chain of the protein. Here's the 5' end. Here's the 3' end. So this side of the DNA grew from 5' to 3'. This one grew from 3' to 5'. And since they're not showing the sugars, you can't really differentiate the 3' from 5' positions. This is not a chemistry class. I'm not going to ever ask you to remember the difference between 5' and 3' positions on a sugar. The second of the 3, messenger RNA. Here's the messenger RNA with its 3-letter codes for the different amino acids. See, here's the code, and here's the codon, and here's the anticodon right here, and it grows, and then there's the attachment site of the amino acid, which would grow from this messenger RNA. Oh, the anticodon, sorry. Where's the third one? There's the third messenger RNA. All right, transcription products used in translation. The ribosomes of prokaryotes and eukaryotes are different in size. Ribosomes found in bacteria, mitochondria, and chloroplasts are of the 70S size, made up of 50S subunits and 30S subunits. I know why, if it's a 50S and a 70, why is it? No, we're not going to go into that great a detail. Eukaryotic ribosomes are 80S in size. They're made up of a 60S subunit and a 40S subunit. What I want you to remember about ribosomal RNA is, or ribosomes, is they are composed of 2 parts, a large unit and a small subunit. A large subunit and a small subunit. The large subunit will bind at the 5' end, or excuse me, the small subunit binds at the 5' end, the large subunit at the 3, and then the large subunit supplies enzymes for making peptide bonds. All right, let's give it a wait for a minute, and I will come back in a few moments and continue this chapter